Hello everyone, welcome to lecture number 7. So today we are going to talk about the two remaining electrophoretic processes. So in previous lecture we talked about particle electrophoresis. So in this lecture we will talk about moving boundary electrophoresis and zone electrophoresis. So let's start with the moving boundary electrophoresis part. Uh, this has some historic significance. So what's that significance? I will come to that later. But let's first try to understand it as a separation technique for protein colloids in free solution. So that means when proteins are dispersed in solution, so obviously they would make a suspended phase. That means basically a type of sol colloidal phase in free solution, and how to separate them from one protein to another protein in a free solution technique. So at that time, means this is a process that was developed almost 70 years ago from today's date. So at that time, uh, this is a glass U-tube, okay, and in this glass U-tube, let's assume that we have a mixture of three types of protein: protein A, protein B, and protein C. This is a mixture, and we want to separate them. And then the glass U-tube is actually it had a stopper, and that stopper is also made of glass. And you see, this is a kind of a stage on the uh, open surface of both the U-shaped tube. And then again we have another two tubular segments. So these are also glass tube. So this one and this one, but they contain buffer. Okay. And then once again, both on the buffer tubes, we have two electrolytic chamber. So these are electrodes, and those electrodes are also dipped in electrolytic buffer. Okay, so the entire process is basically made up for an electrophoretic phenomenon because electricity can be passed, but for the time being you see that these three, you please take a look, this 1, 2, 3 and 4 and 5. So there are 5 segments at the beginning and they are not connected to each other. So you see that the connectivity between the electrode and the mixture of protein is being separated by a buffer medium. Okay, so at the beginning each of these chambers, they do not have any physical contact, they are totally separated, and the electricity cannot be passed through the entire circuit. Okay, so now what happens next is, these are, as I said, that these are the glass stoppers, and both the tubes, the buffer tubes and the electrolytic tubes, so they can move with respect to each other. So at the next phase or next step, these two buffer tubes can be slid towards left this way, and can be connected to the and a connection can be established between the mixture of protein and the electrolytic chambers. So like this. So now we can draw it something like this. So the chambers have been aligned, that is the better word, but still they are separate because there is a glass stopper separating all these chambers. So this is the buffer chamber once again, and this is the protein mixture. Okay, protein A plus B plus C. All right, and then in the third step, what happened that these glass stoppers or these glass doors, they can be removed totally, to making sure the physical contact of all these mixtures. Okay, so at the very end, those glass separators, they are removed, and then at the same time, once those separators are removed, potential is applied across the electrode. Okay, so now the electrophoresis can take place through the tube, and we have a mixture of proteins, right? A plus B plus C, the mixture of proteins. So the thing is, what we can expect. So what we can expect is, so since as I already mentioned before, that different proteins will have different types of zeta potential, electromagnetic potentials at the surface. So depending on their mobility and depending on their zeta potential, the proteins would gradually segregate towards the appropriate electrode. So once these stoppers have been removed, so we can imagine at the beginning, so we had at the very beginning here, so we had all buffer medium here and the protein mixture over here. But once the electrophoresis starts, these buffer zones and the separation between the buffer zone and the actual protein zone would start to vanish. And then what would happen, that some proteins would try to move toward the appropriate electrode. Okay, so just a second. So resuming back, once the electrophoresis process advances, what would be noticed that the proteins, say you have A plus B plus C there, so it's assuming that the A has a better electrophoretic mobility. So the protein A would segregate towards the appropriate electrode first, followed by a mixture of A plus B there, and then we can have say up to here we have a B protein, and then followed by here B plus C, and at the end we have C. So that means the protein A has most electrophoretic mobility, followed by protein B and followed by protein C. So their mixture also, they would lag, means A would be segregated faster than a mixture of A plus B, then B plus C, and lastly we have C. So this method is called moving boundary because, see, there is a density difference between the protein solution, pure protein solution versus the buffer. So therefore, once the pure protein solution segregates, so they do not mix with each other. So that means this buffer layer and this A protein, or A versus B, or pure B, their densities are all different. So since the densities are different, they stack up by some non-miscible or immiscible layers. And due to the electrophoretic effect, so these zones or these bands, they actually move towards the appropriate electrode. So that's why it's called the moving boundary technique, because these are the boundaries. So as if the boundaries are, moving up. So that's why this method is called moving boundary method. And it was difficult to isolate or understand where one layer is ending, or where is the separation between the two layers by naked eye. So the separation of boundaries have been conducted by measuring the refractive index of the respective medium. That means for the respective solution, that means the solution A will have a different refractive index compared to A plus B or compared to the buffer zone. So each of these layers would be differing by refractive index, and there is a technique or there is an instrument by which the refractive index can be measured and through which one can separate a mixture of protein. So at the beginning I told you that these days this moving boundary method has only historical significance. Okay, so why? Let me tell you. So this technique was developed by the scientist Tiselius. So he was the person who first experimentally demonstrated that human blood plasma obtaining in the blood, the human plasma, okay, is consisting of two major types of protein, that is globulin and albumin. And not only that, there are different types of globulins like alpha, beta, etc. So I'm not going to the details of this alpha, beta, gamma, etc. So before the development of moving boundary electrophoresis, it was not possible to separate, isolate, and identify the different types of proteins present in human blood plasma. And this small electrophoretic application gave an immense advantage for the medical treatment. That means the medicine advanced quite a lot with this development or this discovery of moving boundary separation. And you can understand the significance. And therefore, in 1948, okay, so Tiselius was awarded Nobel Prize, okay, for this contribution. And as you can understand, that Nobel Prizes are not being given for unless and until it is a very significant development and has a widespread application for human benefits. So this technique once again is based on electrophoresis. So basically the electrokinetic potential of the proteins suspended in the medium. But these days we do not use this moving boundary method. So this method has been obsolete. So what's the most used or the latest electrophoretic methods, especially for the separation of proteins, DNAs, nucleotides, and other small molecules or small charged polymers? So that method is called the zone electrophoresis. So we will start to discuss the zone electrophoresis, and then we will discuss there that why the zone electrophoresis has replaced the moving boundary methods these days. Okay, so let's clean the board and discuss that. So now we're talking about zone electrophoresis. So this is a small scale analytical technique just like chromatography. And these days zone electrophoresis has replaced the moving boundary electrophoresis for any practical purpose, because this electrophoresis technique is not only simpler and not only cheaper, it is very user friendly. Means anyone with a very minimum amount of training can do that, and most importantly it takes very small amount of sample, and that's the most important point. And using the zone electrophoresis method, so we can separate effectively DNAs, proteins, polypeptides, some other charged macromolecules like charged polymers, some biomolecules, etc. So within zone electrophoresis, there are three major categories. So these are called, one is called the agarose gel electrophoresis. Okay. I should write it carefully. Electrophoresis. Okay. And then we have a technique called SDS page that I will describe later, that what is SDS and what is PAG page. And then the third one is called the capillary zone electrophoresis. Okay. So let's clean up the board and let's start to discuss these different different techniques one by one. But before we start, let me tell you, this agarose gel electrophoresis mainly applies to separate the DNAs, and this SDS page is applied to separate proteins. Okay, and these capillary zone electrophoresis, so they can be used for either proteins and DNAs, but some required modifications are applied. Okay, so let's start with this technique. We talk about agarose gel electrophoresis. So agarose gel electrophoresis, and sometimes this technique is also called the gel electrophoresis itself. So what is agarose gel? So agarose is a substance obtained in a powdered form from a marine algae. It is also known as agar-agar. Okay, so that's a marine algae. From this, agarose is extracted, and this is basically a polysaccharide. Its chemical composition, if we talk about, it's a polysaccharide. And as we studied in a very first or second lecture, that whenever the dispersed phase is liquid and dispersion medium is solid, we can have a gel. Okay, so the same thing happens. So when we disperse water in these agarose medium, we get an agarose gel. So this is so here. So let me give you a brief practical know-how that on how to conduct an agarose gel electrophoresis. So let's follow this. Is a kind of a laboratory instruction mode. That let's take a conical flask, okay, and then there we take this agar or agarose, the powdery substance here, and then we put water here. So therefore we have an H2O layer. Okay, so the water and agarose would not mix up at the beginning. So therefore what we need to do, we need to heat this up. So maybe we can put that entire conical flask in a microwave oven and heat it up for a certain time. And then if we do heat up, then we would get here a viscous liquid, and we can call it, this is the semi-solid gel. So now the liquid phase has been suspended within the solid phase, that's why we get this gel in the form of viscous liquid. So then what we do with this viscous liquid is, then this viscous liquid is actually poured within a tray. So here I am drawing the cross section of the tray. So this is the conical flask, we can imagine. Okay, and we have this viscous liquid, and this viscous liquid is poured because it is not yet solidified. It is still viscous, and it can be drained. Okay, so the liquid can be poured inside the tray. So once we put inside the tray, so the liquid, the viscous liquid will gradually take up the place of the empty tray. And when the liquid is still viscous, okay, means semi-solid, not hardened yet, what we can do here, we actually, one end, we put wedge-like substance, something like this, okay. So this is called gel blocker. So why it is given, it can be clear in the next step. So it has several, it is actually a block, okay, a rectangular block. So if we take a look at the front view of, because this is a side view cross section, if we take a front view of this gel blocker, it would appear something like this. Okay, so something like this. So we put it there, and the liquid within the tray, and we allow it to settle, we allow it to come up to the room, come down to the room temperature, means we allow it to cool down. So the liquid is allowed to cool down. So once the liquid is allowed to cool down, so this semi-solid liquid gets hardened. So it's just like a solidified, looks like a solidified wax. It's pretty solid and it doesn't move within the tray anymore. So after that, what we do is we then forcefully take out that gel blocker from that solidified gel. So therefore, if the gel blocker is put out or taken out, so we have a formation of this kind of hole or ridge within that gel. Okay. So remember, this is just a side view, but if you look at from the frontal side, there will be several such deep or holes created inside this gel, solidified gel. So this works almost like a template. So after that, what we do is we fill up this entire tray with electrolytic buffer. So the same buffer used for the electrophoresis. Okay. Now once we put the electrolytic buffer, then using, because we need a very small amount, so using a micropipette, okay, we actually inject the samples. So the samples are also taken a little bit of electrolytic buffer, and we inject the samples. So here the samples means basically the DNA samples. So we put it inside these holes created inside the gel block. Okay, and once the samples are applied, we introduce electrodes for electrophoresis at the both end. Okay, so maybe this end is positive and maybe this end is negative, and we allow this electrophoresis to take place for several minutes. So now what happens? The DNA samples are there. So as we know that due to the presence of the polyphosphate backbone, means several number of phosphates backbones in the DNA, so DNAs are negatively charged. So these negatively charged particles or the DNA molecules would behave as a charged colloids, that means a salt within the buffer medium. So owing to their electrokinetic mobility, they would come out from the sample well, and they would start to migrate towards the positively charged electrode. Alright, but they would creep on the surface of this gel block. They won't come out freely on the solution. Okay, so therefore the solution here would be free of any kind of convection process, means the buffer medium. So the usage of gel is something like that. The gel is hydrophilic in nature. So it's a polysaccharide molecule basically. So due to the favorable interaction, the DNA molecules would move along the surface. So it would graze along this gel surface like that. So after some time, we stop the electricity and we take out the gel block from the buffer medium, and we actually analyze the gel block. So once we take out, if we look at from the top, so here, so this is the top view. So if we look at from the top, it would look something like this. So here we have these 4 sample wells. Okay, so these are those holes we created using the gel blocker, and inside this we had these samples, DNA sample. So we can write this sample 1, sample 2, sample 3, sample 4. And in all those 4 sample wells, we have DNAs of different sizes, that means different kilobase pairs. So for example, we can think that sample 1 has DNA of 1 kilobase pair, sample 2 has 2, 3, and then sample 4 has 4 kilobase pair DNA. So this, while the electrophoresis was going on, this end was negative and this end was positive. So that therefore the DNAs would be repelled by the negatively charged electrode and will be attracted by this positively charged electrode. So they will start to move there. But the question is, will all these DNAs move equally? The answer is obviously no, because their electrophoretic mobilities would be different. So then what kind of DNA would achieve the maximum migration? So that depends. So we can write the mobility is actually depends on, there is a term, it's called the Z by M ratio or Z charge by mass ratio. So the Zm ratio tells me that in this context Z is referred as the charge on the DNA. So that all the DNAs are negatively charged, and it is said that this charge of all the DNAs are same, all are negative. And the mass, okay. So the smaller the mass, higher the Z by M ratio, and higher would be the electrophoretic mobility. So that means the lighter DNA would move faster. That means at a stipulated amount of time, the lighter DNAs would cover the maximum distance towards the positive electrode. So therefore, once the DNAs move, they actually, so they don't move like a very sharp boundary. So there would be sometimes a streaking happens. A streak would appear, and you can sometimes you can see there is some trailing line of the DNA movement, but the lighter DNAs obviously would move, would travel a greater distance, followed by 2 kilobase pair, followed by the 3 kilobase pairs, so something like that, and lastly the heavier one, so the 4 kilobase pair. So this is just a demonstration. I mean, this is just a representative figure. In actual practical applications, using gel electrophoresis, one can do separation of DNAs involving 500 base pairs to around 30,000 base pairs or 30 kilobase pairs. Okay, even sometimes more than that. So so this entire procedure is called agarose gel electrophoresis, because the gel is used as the grazing medium for the DNAs. And the most importantly, The separation is based on the Z by N ratio. That means the lighter DNA would move faster. So, but before we move on to our next topic, that is the SDS phase, it's just a word of caution on this word Z, because sometimes there is a misconception that Z means the total charge of the DNA. No, Z doesn't mean the total charge of the DNA present. So total charge, by the word total charge, is I mean that there are almost 4 times more number of phosphate groups in 4 kilobase pair DNA than 1 kilobase pairs. Okay, because you know, phosphate, these are the backbones and side chains, so the numbers would be greater. So it does not depend on the total number of negative charges. So the Z is same in all the DNAs, that actually referring to the electrokinetic potential. So that means all those DNAs are negatively charged. So as long as we have intact, undenatured DNA, the electrokinetic potential, in other words, the zeta potential, when they are dispersed in the electrolytic medium, in a certain electrolytic medium, are the same. So therefore, their electrophoretic mobility is only defined by their respective masses. That means the base pairs. The smaller the base pair, smaller the masses, they will move rapidly due to the greater electrophoretic mobility. Okay, so I hope this is clear for you, the Z by U ratio. So now let's move on to the second technique for separation of proteins. It's called SDS phage. So the second method, SDS phage, is almost similar to the GE, that is gel electrophoresis technique, but this is mainly used for the separation of proteins. So where is the difference? So firstly, let's understand the protein. So as I mentioned it before, that proteins are charged uh, polymers, and unlike DNA, they don't have that kind of same negative or positive charge. So each proteins at a certain defined pH, say pH 7.4 or pH 5 or pH 6, at a certain pH, different proteins will have different net charges. So as I mentioned, in that isoelectric point, some proteins are charged less, that means 0. So therefore, we cannot depend on Z by M ratio directly for the proteins. Okay, so therefore different proteins have different masses and different charges, so their Z by M ratios are completely haphazard, and the Z by M ratios can be same for two widely different types of proteins, and they cannot be inseparable. So this GE technique is not applicable for the proteins in the same manner. So there has to be some modifications, some changes. So the changes appear there. So imagine we have a protein over here. So some positive charge, some negative charge, something like that. So this is just a schematic diagram of a protein. Okay, so folded protein. So what happens that we take a protein and we apply a detergent. So this detergent is called SDS, and its full name is sodium dodecyl sulfate, and sometimes it's also known as lauryl sulfate. It's just a detergent molecule, a surfactant. So we will talk more about detergent molecule and surfactant and their actions later in this course. But what happens once this SDS is applied, it is a negatively charged surfactant. So it envelops the protein in such a manner that it may or may not denature the protein, but what would happen that we can imagine the same protein would be now will have a very uniform distribution of negative charge. So the protein the electrical behavior of protein would now almost similar to the DNAs, because of the appearance of these negative charge as they are appearing in the DNA due to the phosphate backbones. Okay, so this SDS mimics the backbones of DNA, phosphate groups in proteins, okay, by just enveloping them. So therefore, we can eliminate the different Z by M, these controversies by having an SDS coating on the proteins. And the rest of the method are exactly the same. So we take the gel block, and here once again we have these 4 samples, 1, 2, 3, 4. So depending on their Z by M, so the proteins would segregate. So here I assume that in lane 1 the lightest protein is there, and in lane 4 the heaviest protein is there, and in lane 2 and lane 3 the same order is following between. So again the proteins are being segregated by Z by M. So once again this M stands for the mass of the peptides or proteins or polypeptides, whatever. But this Z once again, as you can see, this is not the total charge, total negative charges, because if we have a longer and bigger proteins, it will accumulate a lot more SDS, and there will be a lot more number of negative charges. But this Z referring to the electrokinetic potential, the zeta potential at the surface of these protein colloids. So because all of these will have the same zeta potential, just depending on their electrophoretic mobility, which is governed by their masses. So the smaller, means the lighter proteins will move faster towards the, so this side is negative, this side is positive, because the positively charged electron, and we will have an effective protein separation. So now the page. So the thing is, you see, as we are using this SDS, this chemical, the normal gel, that means normal agarose based gel, would not work for protein. So we have to have a different gel medium. So for protein separation, we use a gel. It's called polyacrylamide gel. So that's why this is called PAGE. So P stands for poly and A for acrylamide. So the A stands for acrylamide. So polyacrylamide gel electrophoresis. So this is called gel. And sodium dodecyl sulfate stands for SDS. So this entire thing is called SDS page, means sodium dodecyl sulfate polyacrylamide gel electrophoresis. So if you take a look at majority of the research papers that are getting published from biosciences involving the separation of DNAs and proteins, you will see in almost all of them, they publish a direct picture of these gel blocks. Okay, so you cannot easily visualize this separation of these segregated proteins or DNAs by naked eye. So they has to be stained by some dye, and then some treatments are there, and then some photographs are taken. But to publish a result in a scientific article, these photographs of these gel blocks, they serve as an undeniable proof of the separation of proteins. So these separation techniques are indeed very useful, and they are being widely applied in research and separation techniques involving proteins and DNAs, etc. So that's all about gel and SDS page. So now we would move on to our last electrophoretic method, which is called the capillary zone electrophoresis. So this comes next. So in capillary zone electrophoresis, which is also known as is abbreviated as CZE, we generally establish an electroosmotic flow across the capillary. So the capillary tube is filled with electrolytic buffer, and the electrodes are placed across the two ends of the capillary. So once the electricity is discharged across the capillary, so there will be an electroosmotic flow, because the cations will move towards the cathode and the anions would move towards the anode. So therefore, once the electroosmotic flow is established, within the electrolytic buffer, if we have our charged sol, okay, so it could be some ions, or it could be once again proteins, DNAs, peptides, maybe some charged polymers, some charged macromolecules, whatever, they would get separated depending on their electrophilic mobility. So now here we were talking about pure electrophilic mobility. So, and it would solely defined by their Z by N. So where Z is indicative of their respective zeta potential, and M represents their respective masses. So the Z by M ratio would dictate. So whatever the analytes are, so in which end it will segregate, or it will migrate, whether it will migrate towards anode or it will migrate towards cathode. And depending on their masses or the charge by mass ratio, so there we will have a separation. And while they get separated, their movement or their segregation can be tracked spectroscopically. So this is the reason why the zone capillary zone electrophoresis is also called a free solution electrophoresis. So as you can see that for this method, this may not be the best method for to separate different kinds of proteins. Means the proteins that only differ by the sizes, okay, different number of bases, or means different number of nucleotides, or for just different base pairs of DNA. Is not like that. So there are certain ingredients, there are ions and versus proteins and some DNAs also, where the Z by M ratio, these are distinct and vary significantly. Then actually, and also the anions, they like multivalent anions or multivalent cations. So for those, these capillary zone electrophoresis is one of the simplest and one of the best methods. And sometimes, to have more effective separation, we can have a specially functionalized capillary. Okay, so you know that the capillary is mainly made up of glasses, but sometimes what we can have a surface functionalized capillary like this. So we have a silanol group at the surface. Okay, it means inner surface. So this stands for silicon at one end, and it is attached to the glass surface on the other end, and the silicon is linked with OH. So this is SiOH, and this SiOH group, it has pKa around 2 to 3. So that means if we do this electrophoresis at pH, at the neutral pH 7.4, then these silanol groups will be presented as SiO minus. Okay, so SiOH group will be SiO minus. So therefore, we will have some kind of negative charge. So I will just write it something like this to represent in an easy manner. So now we see the stationary phase would have negative charge. So they would have affinity towards the positively charged analytes. So therefore, the positively charged things would try to get stuck or try to move slowly when it is undergoing electrophoresis. And in other words, the negatively charged analytes would be repelled by this charge and tend to move rapidly, more rapidly towards the appropriate electrode. So here, by doing or by applying surface functionalized capillary tube, one can have the stationary phase to intervene within the capillary zone electrophoresis. So we can write here that here stationary phase intervenes in the capillary zone electrophoresis or in the zone electrophoresis. So the layer means the medium that moves is called the mobile layer or mobile phase, and the phase that remains stationary is called the stationary phase. So these are some very basic techniques of electrophoresis. So we learned about particle electrophoresis, moving boundary electrophoresis for historical significance, and lastly the zone electrophoresis, where we learned gel SDS page and capillary electrophoresis. So through this we just had a very basic working knowledge of what these different kinds of electrophoresis are and how they are applied in different contexts. Okay, with little bit of practical knowledge. So with this lecture, we finished our fundamental properties of colloids in terms of electrical properties. So our electrokinetic properties of colloids has been concluded in this lecture. So in next lecture, lecture number 8, we will deal with the optical properties of the colloids. Okay, so I will see you in the next lecture. Till then, goodbye.